Hi, hello, good morning everyone. I'm Dr. Harika Kishore. On behalf of Vista's Learning, I'm here as your biology educator. Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the live classes, recorded sessions, streaming videos and also study material. Moreover, you're going to get this lecture notes in the form of PDFs. Make sure to get your acquaintance with all the channels that are made available for you, and don't forget to subscribe to the channel Vista's Learning Class 11 and 12. And also, if you find this session interesting and useful, make sure to click on the like as well as share this session with your friends. And also click on the bell icon such that you are going to get all the upcoming video notifications. Saying that, let's begin our today's session. So we know that we are in the series of the sessions and we are discussing about the chapter Molecular Basis of Inheritance, which comes under the unit Genetics and Evolution, right? So today, let's discuss about properties of genetic material. So far, we have discussed about there are so many experiments that were conducted to identify what is the biochemical nature of the genetic material. Starting from the Griffith's transforming principle, Griffith's experiment, and also Avery, MacLeod, McCarthy experiment, followed by the Hershey and Chase experiments. They are a series of the experiments they conducted in order to find out the biochemical nature of the genetic material, right? So here it is clear that the debate between whether it is a protein that is going to act as a genetic material, protein which is made up of amino acids that we already know, DNA which is a double helical structure, RNA so here there is a debate between protein versus DNA as a genetic material. Before that, there was a debate between these three, whether protein is acting as a genetic material, or DNA is acting as a genetic material, or RNA is acting as a genetic material. We know that in majority of the organism, DNA is going to act as a genetic material. And in some of the viruses, like HIV virus and tobacco mosaic virus, TMV viruses, they are some viruses. These are the viruses like retroviruses. These viruses will contain RNA as its genetic material. And after that was Hershey and Chase experiment, it was proven that protein is not the genetic material, but that is DNA. Right? DNA is proved to be acting as a genetic material, and that was an undoubted proof that was given by the experiment conducted by Hershey and Chase. So here is the debate between protein versus DNA as genetic material was unequivocally, undoubtedly, it is resolved from the Hershey-Chase experiment. In the previous session itself, we discussed about this Hershey and Chase experiment where they have conducted an experiment by using bacteriophages. What do you mean by bacteriophages? These are the viruses which can infect bacteria. Literally, bacteria eating, therefore the name bacteriophage came for this type of viruses. So, by taking the radioactive protein and radioactive DNA labeled bacteriophages, Hershey and Chase experiment is able to prove that the DNA is going to act as a genetic material, but not the protein that is going to act as the genetic material, that is hereditary material. So here it become an established fact that it is DNA that acts as a genetic material. Now, though DNA is proved to be genetic material. As we already know, they are the viruses that they form the genetic material. That means they have this genetic material of DNA, of RNA. Now, so here, however, it subsequently became clear that in some viruses, just now we said that, this is very important. In some viruses, we do find RNA is going to act as its genetic material. Example, tobacco mosaic virus, QB bacteriophages, HIV, and even retroviruses, polioviruses. So these are some of the examples of the viruses which contains RNA as its genetic material. Now, answer to some of the questions such as, why DNA is a predominant genetic material? Though RNA is acting as a genetic material, but it is restricted to only small part of organisms, particularly viruses. Whereas majority of the organisms contains DNA as its genetic material. So here, besides being acting as a genetic material, RNA functions as messenger. While we are discussing about molecular basis of the central dogma, where DNA has the ability to produce its own copy, and that process we call it as a replication. This is very important. And also, this DNA transcribes into mRNA, and this process is called as transcription. Now, whatever the information which is present in the mRNA, now it is useful in the synthesis of the protein, and that is called as translation. Replication, transcription, translation. So here, whereas besides RNA acting as a genetic material in the viruses, RNA performs dynamic functions as messenger, see, mRNA, messenger and adapter. While the protein is synthesizing, tRNA attaches to mRNA, which brings the correct amino acid based upon the nucleotides that are present on the mRNA. In this way, the tRNA is acting as a adapter. So here, besides being act as a genetic material, DNA is performing a very important dynamic functions, like it is being acted as a messenger, adapter, even it can act as an enzyme. RNA enzymes, for example, ribozyme. Adapter has to be found from the difference between chemical structure of the two nucleic acids. What are these two nucleic acids? One is RNA and other one is DNA. We do find some chemical structural differences that exist between DNA and RNA. The major difference in the structure of the RNA and DNA, that is, see, DNA has thymine in it, whereas uracil has that means uracil is going to present in the RNA, right? Now, so these are the characteristics, the molecule that can act as a genetic material, whether it is a DNA or RNA, any molecule to that matter, if it wants to act as a genetic material, it has to fulfill some of the criteria that must fulfill following criteria. These are very, very important to understand. These are the list of the criteria that a molecule has to fulfill in order to work, in order to act as a genetic material. In that, the first criteria is It should be able to generate its own replica. In the central dogma, we mentioned that DNA replicates on its own to produce its copy. So here it should be able to generate its replica, copy. That is replication. Now, it should be stable. First criteria, it has to able to produce its own replica. Second criteria, it has to be stable structurally as well as chemically. And the third criteria, it should be provide the scope for slow changes, that is mutation. It should be able to show mutations. It should be able to mutate. Why do you need this mutations? What do you mean by mutations? Mutations are the sudden changes. And that sudden changes has to be happen at a very slower rate. First question, why do we want this mutation? In the previous criteria itself, we said that it has to be stable. If it is stable, it is not going to change. It is not going to show new changes, which is mutation. But here the point is, it has to be stable. But very slowly, it has to show some of the changes in such a way that it is going to adapt to the new characters such that it is playing a very great role. It is contributing for the evolution. Evolution is very, very important. If this evolution is not happening, we might have in the stage of our ancestors itself. But that doesn't happen. That means slowly and slowly, over centuries of time together, it took to adapt a small change, and that is what we are mentioning. These adaptations have to become at a very slower rates. Now, it should be able to express itself in the form of Mendelian characters, laws of inheritance. The characters has to be inherit from one generation to the another generation. So here, if one examines all these criteria, there are 4 criteria, right? So it has to replicate on its own. That means it has to produce its own copy. It has to be stable chemically as well as structurally, and it has to need to show some new changes, that is mutations, which are going to helpful for the evolution. And also the final thing, that what we mentioned, Mendelian characters. It has to express, that is a law of inheritance, law of dominance, law of segregation, what we have learned in the case of Mendelian laws. All this has to happen in such a way that it stores the genetic material and it transforms the genetic information, right? Now, if you examine all this criteria, we need to prove that protein, DNA, RNA. What is the biochemical nature of the genetic material? Each requirement, one by one, there are 4 requirements, right? Because of rules of base pairing and complementary, both the nucleic acids, DNA as well as RNA, complementary base pairing exists in the nucleotides that are present in the 2 strands of the DNA, as well as we can see both the nucleic acids have the ability to produce its own copy. What do you mean by duplication? But the protein can't duplicate itself. So the other molecule in the living systems, such as proteins, fails to fulfill the first criteria itself. So we can eliminate this protein. It is not going to act as a genetic material. It is not going to act as a genetic material. Now, one is DNA and other one is RNA. Both have the ability to duplicate. The genetic material should be stable enough not to change with the different stages of life cycle, with the age, or with the changes in the physiology of the organisms. With the change, with the time, with the different stages of the life cycle, the chemical structure of the genetic material should not be disturbed. It has to be stable enough. And the stability as one of the property of the genetic material was very evident in Griffith's transforming principle. See, while performing his experiment, Griffiths have given the heat treatment to the S strain. And when he combined this heat-killed S strain with R strain, transformation happens in such a way that the non-pathogenic R strain converts into pathogenic form, and therefore he could able to recover the live S strain. So this is all happens because of transforming principle. So here itself the heat which kills the bacteria, at least it does not destroy some of the properties. It does not destroy some of the properties of the genetic material. That is what the non-virulent form is going to convert into virulent form. Now, this now can easily be explained in light of the DNA that the two strands being complementary if separated by any heat treatment or by any conditions, if the two strands of the DNA are separated, but they will come close to each other and forms the complementary and finally result in the formation of the double-stranded again. If the heat treatment is given to the double-stranded helical structure, it is going to separate into two strands, two single strands. These single strands are going to come back to its double-stranded structure when appropriate favorable conditions are provided. When this heat is removed and it is subjected to the normal optimum temperatures, then these two separated single strands are going to combine back to its double-stranded structure. Further, 2' OH group, we know that the pentose sugar which is present in the nucleic acids, DNA and RNA. If you consider RNA, we have ribose which contains at its 2' position OH. This hydroxyl group where the oxygen is missing in the DNA, that is what we call it as a deoxyribonucleic acid. Further, 2', if you look at the 2', we find OH group present at every nucleotide in RNA. Because of the presence of the hydroxyl group at its 2' end, this RNA is reactive. It is reactive. It is not stable, but it is reactive and makes RNA liable and easily degradable. If it is easily degradable, we can say that it is not stable molecule when compared to the stability of the DNA. Just now we mentioned that the pentose sugar which is present in the DNA is devoid of the oxygen atom. Therefore, it is not reactive. It is not reactive means it is stable. Right? Now, RNA is also now known to be act as a catalyst. Just now I said that as it is reactive, it is going to react with the substrate and it is going to speed up the reaction, and that is how it is going to act as a catalyst. and even it is going to act as an enzymes. Example for that is ribozymes. Hence reactive. Therefore, DNA chemically is less reactive. Why it is less reactive? Because it is not having the hydroxyl group at its 2' end, whereas the RNA molecule is having this hydroxyl group at its 2', which makes it more reactive. But the DNA is less reactive and hence structurally more stable when compared to RNA. This is important. The reason is also given. Compared to RNA, DNA is structurally more stable. This is one of the criteria to be act as a genetic material, right? DNA is more stable. Therefore, among the two nucleic acids, RNA and DNA, the DNA is a better genetic material. We're not telling that RNA is not genetic material. It is also genetic material in some of the viruses. But DNA is going to be act as a better genetic material because it is having an extra stability because of the absence of hydroxyl group at the 2' end. It has only hydrogen. Now, in fact, the presence of thymine in the place of uracil. Uracil is present in RNA. Thymine is present in DNA. And the presence of this thymine is also going to give an additional stability to the DNA. Now, both DNA and RNA are able to mutate. It is one of the criteria. Though it is stable, DNA is stable and RNA is less stable. But both are going to show their ability to show the mutations, to undergo mutations. In fact, RNA being unstable, just now we mentioned, it shows more mutations. It shows the mutations at a very faster rate. Now, consequently, viruses, those which are having this RNA as its genetic material, as the viruses will have a shorter lifespan, this RNA will mutate at a very faster rates and therefore evolve faster. If you look at the lifespan of the viruses, they have very shorter lifespans. And therefore, we don't have that, that means viruses don't have that much time to be very stable and shows mutations are very slower rates, which normally happens with the DNA. We mentioned that DNA is very stable, but though it is very stable, it also have the ability to show the mutations at a very faster rate. It will be really very beneficial when the lifespan is very more. But that is not the case in the viruses. Viruses will have very shorter lifespan. And therefore, it has to grow faster, show the mutations faster, and evolve faster. That is what made possible by the RNA taking it as a genetic material. RNA can directly code for the synthesis of proteins. So this is one of the factor we can say. DNA first has to transcribe into mRNA, then protein. If you look at the RNA is directly coding for the synthesis of the proteins. It is not depending upon the DNA. If you look at the DNA first has to transcribe to RNA, then it is going to translate into protein. That means RNA depends on the DNA depends on the RNA, that means indirectly, whereas RNA directly translates into proteins. So RNA can directly code for the synthesis of the proteins, hence can easily express the characters. However, just now we mentioned, this is a molecular biology of central dogma. DNA, however, depends. It depends on the RNA to be translated into protein for the synthesis of protein. right? Now, the protein synthesizing machinery has evolved around RNA, because whatever the information that is present in the DNA is first transcribed into mRNA. Now the mRNA transmission of the information takes place by the RNA. That is what synthesis of this protein molecules evolves with the RNA molecule. The above discussion indicates that both RNA and DNA functions as a genetic material. This is clear up to now. But DNA being more stable is preferred for, this is important, as the DNA molecule is stable. DNA will be useful for storage of genetic information. This is very, very important. Whereas for the transmission of genetic information, RNA is better. So RNA is better for transmission, where from RNA information is useful to place the amino acids. That is, a sequence of the amino acids is decided by the information which is carried by the mRNA. But the RNA got its information from DNA. So for storage, DNA is better. For transmission, transfer of whatever the genetic information, RNA is better. These 2 points are very, very important. Now, moving on to the RNA world. RNA was the first genetic material. There is now enough evidence to suggest that essential life processes like metabolism. Metabolism is all about the anabolism and catabolism, how the food what we are taking is going to convert into energy. That means breaking of the food, that is catabolism, and formation of the energy, that is anabolism. Both destructive, whereas the constructive process together we call it as a metabolism. And translation, just now we said that how the mRNA produces protein by attaching amino acids. These are all amino acids, and this process is called as translation. Translation, right? While this is happening, the mRNA, while the mRNA is producing transcript, and it will undergo the process of splicing. Splicing, where we have the combinations of introns and exons. Introns, exons. Introns, exons. Splicing is the process where it involves in the removal of the introns and addition of exons. See, exon, exon, exon. These introns will get removed, and this will finally attach to form mRNA, and that is called as splicing. So here, introns are the non-coding, exons are the coding sections of the RNA. RNA used to act as a genetic material as well as catalyst because it is reactive. There are some important biochemical reactions in living systems that are catalyzed by the RNA catalyst. And even RNA is going to act as an enzyme. Example for that is ribozymes. And not by the protein enzymes. They are catalyzed by the RNA enzymes. That means RNA, besides being acting as a genetic material in most of the viruses, it is also acting as an adapter. It is also acting as a messenger. It is also acting as a catalyst. Now, but RNA being a catalyst, it is more reactive and hence definitely unstable. Therefore, DNA has evolved from RNA. See, we are mentioning that DNA is stable. DNA is better to act as a genetic material when compared to the RNA. DNA will be acting as a genetic material in majority of the organisms. But this DNA has evolved from RNA itself with the chemical modifications that makes it more stable. RNA molecule undergoes some of the chemical changes such that it is going to be more stable, and that more stable form of the RNA is nothing but DNA, because DNA has evolved, it has come up from the RNA. Now, DNA being double-stranded, and having complementary strands, that means base pairing, where purines forms a base pairing with the pyrimidines, and the pyrimidines forms the base pairing with the purines, like using hydrogen bonds. Already we have mentioned about it, like purines, these are purines, and these are pyrimidines. So bonding between them will be this is base pairing. Adenine pairs with thymine, thymine pairs with adenine. Similarly, cytosine pairs with guanine, and guanine pairs with cytosine by forming 3 hydrogen bonds. And here we find 2 hydrogen bonds. Further restricts changes by evolving a process of repair. This process of repair is an evolutionary that we do find in the case of DNA. That is what we call it as a DNA repair. Because of the presence of DNA repair, it maintains its stability. Whenever there is a small change, that it will repair by itself such that it can maintain the stability of the molecule. Now, so here, while proposing the double helical structure of the DNA, Watson and Crick immediately proposed a scheme for replication of DNA. How this DNA is replicating? This double-stranded DNA will separate into single strands, and this single strands will act as a template for forming a new strand. This is how the new strand will form by taking one of the strand as the template. To quote the original statement, that is as follows: it has not escaped our notice that the specific pairing, base pairing, we have postulated immediately suggests a possible copying mechanism. Here is a copying mechanism. As the base pairing is followed, definitely if the purine is present, if the adenine is present, thymine will come to the daughter strand. Copying mechanism for the genetic material. Now, this is how we can see double-stranded DNA first separates off to form single strand. Using the nucleotides which are present on this mother strand, what we call it as a template strand, the daughter strands keeps on forming, which leads to the double helical structure again. The scheme suggests that 2 strands would separate Here is a two strands, double helical structure, double stranded structure. It will first separate to form single strands. These are double helical structure, that is double stranded. Now it is going to separate into single strands. This is one single strand and this is another single strand, right? And act as a template. These strands, single strands, are going to act as a template for the synthesis of new complementary strands. Just now we said that adenine pairs with thymine, thymine pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine. Using this complementary system, see for example, here cytosine we have. Cytosine is present on the mother, that is template strand. Then the daughter strand will have guanine on it. Adenine, thymine, thymine, adenine, guanine, cytosine. Simple. So after the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. See, look at here. This is the parent DNA. Parent double-stranded DNA is going to separate into single strands. And this red, which is represented with the red color, these red strands are going to act as a template for the newly formed dotted strand, which is represented in purple color. So here, if you look at, these are going to separate as a daughter DNA. If you take the daughter DNA, it will be having one strand coming from parent DNA, and the other strand which is coming newly formed, it will be daughter. So DNA molecule would have one parental, this is parental, and one newly synthesized strand. This is newly synthesized strand. So because of this reason, we are calling it as a semi-conservative. Semi-conservative because one strand is completely coming from the parent DNA. The scheme was termed as semi-conservative DNA replication. Semi-conservative DNA replication. Now, so this is it. So in the present session, we discussed about the criteria to be fulfilled by the genetic material, and also we have discussed about the RNA. We have initiated the replication process. In the further session, we are going to study in detail about the replication of DNA. Until then, keep learning with We Learning. Thank you.